today's conference. Today, I would like to present our research transgalin to a potential paclitaxel-resistant target and the reversal of drug resistance. Okay, I'm working in the pharmacy department of first affiliated hospital of Xi'an University. And our hospital is one of the largest medical institutions in the northwest of China. And it possesses about 36 different clinical departments and 3,000 beds. And we help patients resolve their medical problems. In our pharmacy department, the daily work contains ward-round consultation, medication education, prescription audit, therapeutic drug monitoring, and genetic testing. Additionally, the routine work of drug dispensing, drug supply, and drug preparation are necessary to patients. In our department, the major focuses of research includes individualized chemotherapy of antimicrobial agents, clinical evaluation of drugs, and cancer drug resistance and tumor angiogenesis. Our team mainly engaged in the research of breast cancer drug resistance. Okay, now I'm introducing our ongoing research transgalin to a potential paclitaxel-resistant target and the reversal of drug resistance. As is well known, cancer drug resistance is very complicated, and more than 90% of patients who died of cancers had developed a certain degree of multi-drug resistance. And MDR includes endogenous resistance and acquired resistance. Resistance mechanism is not fully understood, and it involves multi-targets and multi-factors such as upregulations of transport proteins, alteration of enzymes, apoptosis associated factors, and DNA damage repair. So it's extremely urgent to gain further insight into the molecule. And urgent to gain further insight into the molecular mechanism of MDR and identify new biological targets for predicting sensitivity of chemotherapeutics. First of all, we established paclitaxel-resistant breast cancer, and it reached a hair resistant by dictating its sensitivity to paclitaxel, and the resistance factor was 115. We further studied its resistance and found that it possesses a cross-resistance to other chemotherapy drugs. Comparing the sensitive cells, mRNA and protein expressions of ATP binding cassette transporter, and such as MDR1, MRP1, and BCRP were all upregulated in the resistant cells. It had an MDR phenotype to further search for resistant targets. Then, using 2DE and mass spectrometry, we successfully identified 17 different proteins between sensitive and resistant cells. As shown in the table, we found transgallin-2 was significantly overexpressed in resistant cells, and it was expected to potential drug-resistant target. After that, we selected 5 proteins to validate their mRNA and protein expressions. Because their levels were different between sensitive and resistant cells, and they played important roles in tumorigenesis, cell proliferation, invasion, and apoptosis. As shown in picture, the protein and mRNA expressions of transgallin-2 were also significantly overexpressed in the resistant cells. Transgallin-2 is a member of the KL modeling family involved in the cytoskeleton formation and found to be closely involved in the to be closely associated with tumorigenesis. To explore the function of transgallin-2 in the MDR, we inhibited its level with small interfering RNA. The study showed that knockdown of transgallin-2 could decrease expressions of ABC transporter and dual luciferase reporter gene, as it proved transgallin-2 could regulate the promoter activities of ABC transporter. This implied transgallin-2 played an important role in the drug resistance. In the following study, we investigated the resistant mechanism induced by transgallin-2. Comparing with sensitive cells, PI3K-AKT signaling pathway was activated in the resistant cells. Downregulated P10 and upregulated P-AKT was observed in the resistant cells. Furthermore, the downstream pro-survival molecule BCL2 and BCL2 was upregulated, and the pro-apoptotic molecule BAX was downregulated in the resistant cells. And it was shown that cleave, Kisbis9, Kisbis3, and PRP were all downregulated in the resistant cells and inhibiting their pro-apoptotic function. Therefore, the high expression of transgallin-2 activated PI3K-AKT pathway, inhibited mitochondrial apoptosis pathway, and prevented cells apoptosis. On the contrary, the suppression of transgallin-2 immensely augmented P10 expression, resulting in inactivation of PI3K-AKT pathway and revitalized the mitochondrial apoptosis pathway. Based on the resistant target, transgallin-2, we screened effect reversal agent using MTTSC, and it was found that salvinoleic acid A enhanced the sensitivity of resistant cells to paclitaxel and increased resistant cells apoptosis. While SA is a major phenolic active component extracted from salvia, methyl possesses extensive pharmacological activities. In the reversal study, and three different concentrations of SAA, both of protein and gene expressions of ABC transporters were decreased remarkably in resistant cells. It suggested that SAA could reverse paclitaxel resistance of breast cancer by reducing ABC transporter expressions to sensitize resistant cells to paclitaxel. Similarly, the mechanism of SAA enhanced reversed paclitaxel resistance may involve in targeting transgallin-2 to increase p10 expression, cause PI3K-AKT pathway inactivation, and promote mitochondrial apoptotic pathway activation. In conclusion, transgallin-2 was responsible for paclitaxel resistance in breast cancer by activating the PI3K-AKT pathway and advancing cell proliferation. It is verified that SAA at non-toxic concentrations can enhance CAMP sensitivity of paclitaxel in human breast cancer cells by targeting transgallin-2 to accelerate p10 expression, inactivate PI3K-AKT pathway, and increase cells apoptotic. The suppression of transgallin-2 could be an attractive strategy to improve current therapeutic regimes. This preclinical evaluation of a rational combination of SAA and paclitaxel in relevant in vitro model also provides a molecular basis for the new design of combination of treatments for breast cancer. Here are some articles published by our team in recent years. This photo is our research team, and in the middle is our teacher. So thanks for your attention. Welcome to China. Welcome to Xi'an. Thanks very much.